Good afternoon, children. We are going to learn today something connected to cell cycle and also the thing called chromatin and its changes during the cell cycle. Today is 31st May 2021 and it is almost now about 2:30 p.m. Alright, so we are going to have a recap of cell cycle. You know that a cell which contains nucleus inside and many nuclear material called organelles. All these things in a resting cell, a cell which is not active or resting cell not dividing, and this enters into what's called the cell cycle. Cell cycle. That means this cell now prepares itself to become two daughter cells, or what you call cell division or cell reproduction. We shall start with that, and then we come back to the chromatin and its importance and changes in the system. Now in the cell cycle, if you want to show, I will try to say how it happens. Now please see how you can write it. If you are a student of 9th or 10th or 11th, this may be helpful to you. There will be one phase to begin with. You call that G1 phase, and then there is another phase that is very important one, and particularly this particular phase, what we are now referring to is the second phase, and in the second phase this is called S phase, and then comes the G2 phase of the cell cycle, and then comes the division of what you call, either it may be mitosis or it may be meiosis. Any one of these two may happen, and ultimately we get the cell division or cell divides into daughter cells. Cell divides. This will be a kind of a cell cycle. What happens we shall see. In the G phase, all the cellular contents here, whatever contents we have, they get duplicated. They get duplicated except the chromosomal material, or except the chromatin, except that everything gets duplicated here. And then in the S phase, when it comes to S phase, in the S phase, only the nuclear material that is chromatin now divides and becomes duplicate. Now it becomes duplicated. That's called the S phase. And then comes to G2 phase. In the G2 phase, what happens is there will be it's just checking. The cell checks whether everything is all right or not. That is the G2 phase, and then it enters into what you call the kinetic phase, where either mitosis takes place or meiosis takes place. In each case, there will be some differences. Ultimately, we come to what's called the cell division or cell divides. You get either daughter cells, two daughter cells we get it, or you get the gametes. Either of the two we get it. Either we get gametes or we get the daughter cells, which will grow into tissues. So this is in short a cell cycle. That means before the cells divide, lot of internal changes will be happening, and that is called cell cycle. You might have studied this in your classes. Maybe I just made it for you a revision. So let's remember a cell after the resting stage divides into two daughter cells, or it divides into four gametes. Either of the two can happen. In either way, there will be one phase called the G1 phase, where all the cellular contents like mitochondria, plastids, whatever it may be, if it is a plant cell, something different, animal cell different, they get duplicated. The next in this stage, or the G1 phase, nuclear material will not duplicate. That will not duplicate. Only in the S phase, that is synthetic phase, the nuclear material divides into two. Duplication takes place. If there is one chromosome, it becomes two chromosomes. If there are 23 chromosomes, it becomes 46 chromosomes. The normal number in a cell will be called 2x or 2n. That means one from the one parent and the second one from the second parent. This is called 2x. So even this will become double. It becomes 2, this becomes 2 in the synthetic phase. That's the important thing you must remember. And then in the G2 phase, there will be a check. The cell checks everything, whether there is any error or duplication complete or not, like that. And finally, the cell enters into stage called mitosis, where thread-like structures like the chromosomes will appear, and that will go to daughter cells, or they may go to stage called meiosis, where reduction division takes place and they become gametes. So this you may study at a later stage. If you have studied, try to recollect it. Now, after having learned that every cell has a cell cycle, and in the cell cycle there will be large number of changes. Now we shall see how the chromatin behaves. Now again I will just try to draw a small diagram. Let's say that this is a cell, imagine, and in this cell there will be a central portion called the nucleus, and this nucleus will contain a large number of materials. One of them is called the genetic material called the chromatin. Chromatin is a complex material. It contains DNA, our genetic material. It also contains some histamines, and it also contains some proteins. It's a complex. So if you just study this particular thing, chromatin, let's say we take the chromatin out, and the chromatin may look like that. A long thread, a long spiral thread, one turn, two turn, three turns, like that. Many turns will be there. That will be a long thread called the chromatin. And this chromatin, I told you, contains DNA. It is covered by several things. So DNA will be seen inside the chromatin. DNA will be seen here. That also will be highly twist will be there, spirally to be there, and that means the cell now unfolds the chromatin. Now in this chromatin, if you just look at it, there may be small places. Let me mark it in a red color so that it will be easy for you to remember. There will be small segments like that. There will be one segment, imagine, and there may be another segment here, another small piece, and there may be another small piece here. Like that, there are certain areas in the chromatin which we can call them as genes. Genes. Now here I have marked 1, 2, 3 colors. Three genes I have marked here. So every chromatin may contain several genes. Now, what is the gene? We shall try to understand now. Now, this chromatin material divides itself into smaller pieces like that. It depends upon the species of the organism. It divides. In human beings, it will be 23 pairs. It divides. And as we said earlier, that will be having its own duplicate material inside that. That will be duplicated chromosome. And these chromosomes now will be there in the cell. Now, if you take a small section of that particular thing, I'm going to rub it off now. Let's take a small segment of the DNA and find out how it looks like. The DNA, you say that it may form like that. It's a long chain, and this long chain may be having several small twists and turns like that. It is not very simple to understand. It is the Watson and Crick who said that the chromosomes contain material called DNA. So now like that, it's a long thread-like structure containing a large number of sticks, and this is the DNA. And inside they found that there will be small molecules. Molecules are there inside this. These molecules are called nucleotides. Nucleotides. So each strand, this is called strand 1, let's say A, B, there are two strands, two parts are there, and there will be two parts, and each one will contain nucleotide, and they form the DNA. That is the DNA structure. Now, we will go straightly a little more details into that one. Let us draw a little more detailed diagram. Let's take up a section of DNA. That's a section of DNA, let's say. And this section of DNA, firstly, will contain one like that. These are the materials present inside the DNA, and I will just marking these things so that you know that there will be each one of them. Let's say these are called the nuclear bases. I told you this is one nucleotide, another nucleotide, another nucleotide, this another nucleotide. So there will be two nucleotides together and they form a base pair. There may be some more things like that which will be in a different way. Well, I am just marking them so that you understand that. This is another type of nucleotides. So there will be, I have used 1, 2, 3, 4 colors indicating there will be 4 types of nucleotides, and each one of them we can call them by name. One of them is called adenine base, another is called thymine, another is called cytosine, another is called guanine. So these are the 4 types of bases that will be seen throughout the length of the DNA. Now DNA has maybe a long thread. If you take up only one segment of the DNA, which I wrote to you earlier, if you say this is a very long thread of DNA, if you take only one small portion of it, that small portion is called the gene, and that will contain a definite number of bases, and those bases will be adenine base, and that will go with the thymine, and cytosine goes with the guanine. So this is the way they pair. It's called the base pair. Like that, thousands of base pairs may be there. They are called sequences. There will be a large number of sequences. Supposing we say this is one gene, that is one segment of DNA, one segment of DNA. This DNA segment may contain hundreds of sequences of bases. Now, each gene may code for one particular type of protein or enzymes. Now, that details we can study a little later on. Now, I told you that each of these, like adenine, thymine, cytosine, guanine, they are all the bases. They are seen in the whole DNA structure. Now, if you just want to study or remember how these things will remain there. Now, each nucleotide will contain one phosphate. It's called the phosphate group. It has got a phosphate group. The phosphate group now will be joining. Phosphate will be joining with another group. That group is the sugar, pentose sugar, and this pentose sugar will be connected to a base, nuclear base. So there is one nucleotide. This is one nucleotide. So this is phosphate, this is sugar, pentose sugar, and this is base. Now this base may have either two pyrrole rings, they are called pyrrole rings, or it may contain two rings. Let me write to that one also. It may contain one. So it may contain two rings or another one may contain only one ring. Instead of this one, there will be only one ring like that. One ring. So the base may be either made up of two rings, they are called pyrrole rings, or it may be made of one ring. So the nucleotide base pairs will differ, and the gene which is nothing but a small segment of the DNA containing definite number of nuclear bases, they will code for one particular type of a protein. And how protein is formed is another question that will have to be studied in what we call protein synthesis in using mRNA, using ribosomes. That we can study in the next particular session. So now let me recall to you right now. We studied cell cycle. Cell cycle, we said we will have four important phases. G1 phase, where except the nucleus everything will be duplicated. Synthetic phase, where the nuclear material doubles. Then the G2 phase, where the errors are checked and then enters into cytokinesis and kinetokinesis. That means the either mitosis or it may become the meiotic division. Ultimately, new cells are formed. The cells may be somatic cells, body cells, or they may be gamete cells. When you study them, you find there will be difference in the nuclear material or difference in the number of chromosomes present in the body cells and also in the gametes. That's the first thing we studied. The second one, we studied the chromatin. Chromatin is a very, very long thread, and that will be almost having several turns and coils. That internally it contains DNA material, and it is covered over by histones and proteins. and that chromatin starts dividing during the cell cycle, and they divide into number of smaller segments called the chromosomes. Now each chromosome, when you look at each chromosome may contain smaller segments inside. Each segment may produce a protein. One small segment of DNA is called the gene. So a chromosome may contain several genes. I am told that the one of the chromosomes which controls the eye color in the human body, in the human beings, may contain 20,000 genes, and genes interact and they produce different types of materials. So those who are interested in studying the genetics may try to read up these things. I will end up here. Thank you very much.